Barsan is professor and former chair of emergency medicine at the University of Michigan Medical Center. He is the principal investigator for the Neurologic Emergencies Treatment Trials Network, and he was also an investigator in the NINDS TPA study back in the early 1990s and instrumental in its design. Without further ado, I'm going to pull his presentation up, and we will let him get started. He will need to remember to unpause his camera and unmute his microphone. Oh, look, he's already there.
I did them both.
So,
I did them both. I'm way ahead.
all right. Are you unmuted?
Yes. I should be unmuted. Can you hear me?
So I'm going to get your. Yep. There it goes. Then we'll go back to my presentation when he's done.
Okay.
Go back to my presentation when he's done.
Oh, I don't think we're at the beginning here, Will. Great, here we are. Okay, I'm assuming you can hear me now. So it's a real pleasure for me to be the kickoff speaker for the clinical trials course. And what I'm going to cover is just, after being involved in doing clinical trials for over 30 years, I think some things that have occurred to me in terms of issues with our process for translational research, and I'm going to try to give some comments on that using neuroprotection as sort of an example. But behind all this, I think the question is that, a lot of times I think we've gotten into problems in our investigations because we haven't started by asking good questions. So really, the title of this is Asking Good Clinical Trial Questions in Translational Research, but most of what I'm going to talk about is really a result of not asking good clinical questions for the most part. So my only disclosures are funding from NINDS and FDA. And the objectives for this talk are going to be to understand the issues regarding translational pathway of treatments into established therapies. And again, as I mentioned, using neuroprotection as an example. And I think we want to talk a little bit about how sometimes you can get started on the wrong track. And sometimes these early phase trials can, rather than trying to answer questions, a lot of times we can actually propagate uncertainty sometimes with these early questions if we don't ask them right. And I'm going to talk a little bit about the current challenges in translational pathway, again using neuroprotection as an example. So in terms of crafting a good clinical trial question, in general you should just have one main question. I think everybody's aware of that you can only choose one primary outcome, and so you've got to ask one question. And it can be, you could do it very simply and just your question would be, is X better than Y? Maybe better than that would be, does X improve a patient-oriented outcome compared to Y when administered to this population for this long at this dose, which is a little more, it's a lot more substance to that question than the original one. And then you can say, well, could the question have been even better? What dose, schedule, and population would hold the most promise for determining that treatment X could be proven better than treatment Y in a phase 3 trial? And I think one of the things that we have, that I've become learned a lot, I think, in the last several years, and I think true for a lot of other people, is that a lot of times when we're designing trials and coming up with clinical questions, sometimes we're trying to eliminate uncertainty and say, well, maybe we're not entirely sure of this dose or whatever, but we're going to go with it. And, we're going to be certain that that's what we want to do. And I think more and more, I think we're coming to the realization that recognizing that uncertainty and actually embracing that uncertainty is a better idea than ignoring it. The other thing that I think we've been using a lot is that trying to say, what would I have done differently if the trial ends up failing? So when you're designing the trial and you think you've got the right dose and you think you've got the right population or whatever, and you think you've got a plan that's pretty good and you're asking the right question, I think then you have to ask yourself, okay, if this trial fails, what do I wish I would have done differently? And if it's a drug trial, for example, you might have said, oh, well, it failed. I would have used a different dose. I would have used a higher dose. I would have treated for longer. I would have treated earlier in the course of the disease. And I think this recognizing and embracing uncertainty, if you address these things at the beginning when you're designing it, I think you end up with a much better trial in the end, as opposed to trying to pretend like you don't have any uncertainty. So first question is going to come up here, and we'll let people go ahead and answer these, and I'll continue when we got the answer. So what is a failed clinical trial? I talk about having failed trials. Is it a trial with a non-significant p-value in the primary outcome? So in other words, you failed on your hypothesis. Is it a trial that doesn't provide the clinical and scientific community with understanding the next steps? Or is it a trial that enrolls fully and provides actionable results, but only in a small, discrete subset of patients? So everybody, if you haven't voted, please go ahead and vote. Looks like we didn't fool anybody on that one. Yes, obviously it's the trial that really doesn't help you to understand what's going on with that problem. So neuroprotection, which I'm going to use as an example, can really apply to any acquired neurological condition. And we all know that there's an injury cascade that's started by either ischemia or trauma or whatever, and you can hopefully, the idea is to try to impede the injury cascade by targeting one or multiple pathways. It can apply to a lot of diseases: stroke, TBI, global ischemia, and among these I'm going to use stroke and TBI most because they've been the most studied areas. So this is just sort of a graph showing the timeline of molecular pathways that are activated after acute stroke, using stroke as an example, and you can see that most of the action is happening very early. I mean, within 2 to 4 hours, all of these pathways are getting activated rather, and things are happening. The time, and then if you're talking about reperfusion injury with either stroke or cardiac arrest, this is the timeline for activation of molecular pathways that are active after reperfusion. And again, a lot of the action happens very early on. So one of the things that we've got to face up with in neuroprotection is that neuroprotection has been a drastic failure. We have done at least 20 late phase 2 or phase 3 trials for TBI. All have failed to show a benefit. In other words, none have worked. More than 130. This is despite the fact that more than 130 monotherapies look good in preclinical work, but nothing's worked in phase 2 or 3. And the most recent example of that is progesterone for TBI, which we did in the net. That seemed to be very effective in animal trials. There was a phase 2 trial that indicated that it was going to be, made everybody very encouraged, but actually failed in the phase 3. Ischemic stroke, 15 drugs have advanced to phase 3 trial testing. All have failed in ischemic stroke, and so we've got basically a pretty bad track record. We've looked at multiple classes of drugs, and this is just some of them, and really none of them have worked. So what are some of the reasons why they might have failed? A lot of people in the past have pointed to inadequate preclinical testing. There are some STAIR criteria, which were criteria for preclinical testing for stroke, which was done in an academic industry roundtable. And I'll talk a little bit about the STAIR criteria. We've also had problems with inadequate clinical testing. And then in some drugs that might have a narrow therapeutic index, that you really can't get the dose that you need to be, that is active because of toxicity. So these are the STAIR recommendations, the original ones that were developed for preclinical stroke development. And I'm not going to read every one of these, but you can look at these. Basically, you're talking about looking at permanent and temporary occlusion in different models, getting an adequate dose response, that you should do monitoring and blinding, etc. And these were followed for several neuroprotective agents, and what happened was they all failed, despite the fact that people use these STAIR recommendations for their preclinical work. So in response to that, there were modified STAIR criteria, and these are the modifications that were made, and they're in bold. And the first one in number 2 there sounds pretty basic document the drug gets into the brain. Well, sometimes that wasn't done, and you can look at the other ones there too. So we keep trying to make these better to say, is there a way you can do preclinical trials that will actually predict what's going to be successful in the clinical arena? We're not there yet. So clinical trials, failures in stroke, a lot of reasons for them. A lot of times tested too late after stroke onset, inadequate sample size. If you're looking for a modest treatment effect and you just don't have the sample size, you're not going to see it. Sometimes the answers get diluted by the fact we have too many mild patients or too many very severe stroke patients. The mild ones are going to get better on their own. The bad ones are going to do bad no matter what you do. And so trying to detect a treatment effect can be difficult. And then again, side effects can also be a limiting factor in some of these. So we'll go ahead to question number 2. This one's probably even less tricky than question number 1. So analogs to the STAIR criteria exist for other neurological conditions, including Alzheimer's, dementia, and ALS. True, false, or C? Anybody? All right, when you think you've got enough, are you done? Okay. So we've got, good. So we got about split between most of the people in B and C, and the correct answer is C. The NINDS has developed a transparency and reporting guidance that would be good to read. And in fact, these STAIR type criteria do exist for other neurological conditions. So if you do research in these, particularly in preclinical work, it would be, and even if you're doing clinical work, it'd be very helpful to be familiar with those. Other reason for clinical trial failures in stroke, sometimes there's too many lacunar strokes, and some drugs don't have any evidence for working in white matter. Lack of penumbral imaging in patients enrolled at later time points, and this is a really tricky one in any of the trials, and that is that if you're putting patients in a neuroprotective trial and some are getting TPA, the effect of any neuroprotective agent might get totally overwhelmed by the effect of reperfusion with TPA, and so that makes it very, very difficult to measure some of these effects of neuroprotectants in stroke. So what's our typical model? What do we usually do? If we're looking at drug discovery and we're looking at how drugs and devices move through a pattern when we're trying to figure out what works. So initially we got discovery research into pathways, mechanisms, looking at what receptors are activated by what. And then we've got preclinical animal models of disease. You've got the threat occlusion stroke model, different things like that you might use that you think mimic the clinical condition. And then when we get to people, we do phase 1 testing for toxicity and pharmacokinetics. We do phase 2 trials for safety and, quote, potential efficacy, and then phase 3 trials. So there's a restriction of flexibility in drug and device development. I'm going to sort of go over this. So in either drug, we do phase 1 study, and devices, we do bench work. We make some decisions, we go on to phase 2 or early clinical testing. We make more decisions, and then for the drugs, we go to phase 3. For devices, we go to trials that are not really efficacy trials, but they really confirm that the device is doing something. So if you've got a device that's supposed to give you clean arteries, if it works and it gives you clean arteries, then that device works. It doesn't mean that it's effective in improving outcome. It just means that it does what it says it's going to do. So the steps in traditional drug development and sort of how we approach those, usually in phase 1 we're talking about not large number of patients. Can often be first use in humans, and you're really looking for just general toxicity and dosing info. Phase 2, and this is where I think most of our problems arise. We do 1 to a few hundreds of patients with the target illness. We get some initial information on dose-response relationship, and we hope, proof of concept, get some idea of what some of the side effects might be. And then when we do these phase 2, if we don't have horrible side effects, and it looks like the treatment is probably safe, we will move on to these phase 3 trials to try to confirm superiority. And you usually need 2 adequate and well-controlled trials to demonstrate that. So what are some of the challenges? Sometimes in phase 2, a wide range of dose strengths may be possible best choices. And I'll use the TPA stroke studies as a great example, because I was involved in the TPA pilot study where we were looking at a dose escalation of doses of TPA in stroke. What we were able to show is that we got to a dose that was high that we knew was dangerous. Okay, so above about 0.9 milligrams per kilogram. But we really had no idea what dose was the most effective in terms of reperfusing arteries. So we, 0.6 milligrams per kilogram was safe, 0.4 was safe. They seemed to work just as well, but it was small numbers of patients. There were only 75 patients in the whole dose escalation trial. But we basically, chose a dose at the end of that to say, okay, this is the one we think is best, and mainly because we thought it was the highest dose that's safe. A lot of times in phase 2 trials, we don't consider combination treatments or different durations of treatments. So again, I'll use TPA trial for example. We did a 1-hour infusion. Would it have been better if we would have front-loaded the dose? Would it have been better if the infusion was longer lasting? We don't know. We never tried and have no idea. Maybe that's a much better way to do it than the way we did it, but we don't know that because that isn't the way we did it. You may not know really the right patient population. So, is this just as effective in younger people, older people, embolic strokes? It's really unclear to know that. And most phase 2 studies are really, when you look at them, most are actually underpowered phase 3 trials. So they're really looking at not a large enough number of patients to even begin to get at efficacy. Mostly getting some idea of safety and maybe a trend towards one thing or another. But really, they're very underpowered phase 3 trials. And in the end, I think a lot of times we don't really learn a lot from them. And we usually don't compare different agents successful in phase 2. So you might have 3 drugs, drug A, B, and C, that have all gone through phase 2 trial testing. Of course, all different, right? Different inclusions, different exclusions. And they all seem reasonably safe and maybe have some reason to think they might be efficacious. Which one do you use for a phase 3 trial? Because you have no way of comparing them to each other. They've all been tested differently. And so trying to know which ones you move forward into phase 3 is really impossible. But the challenges in traditional approach with phase 3 is we still don't know the right dose or timing often. And TPA study is an example. So we said we think this 0.9 is the best dose. We're going to go with it. We didn't really know if that was the best dose. We really don't know what to expect in the control arm, and this can be a big problem. If you're assuming that the control arm, for example, is going to have a 50% incidence of ending up normally no matter what you do, and if it turns out that when you do the study, actually 75% of the controls end up normal, you got a big problem in terms of trying to show the difference. Sometimes we don't know the right population to study, and sometimes we don't know about rarer side effects, and you just need more patients. So despite that, despite all this uncertainty, the right dose, what to expect in the control group, we don't know the right population, our traditional approach says you have to completely define all these trial characteristics when you do a phase 3 patient. So in other words, when we designed the TPA trial, even though we weren't sure we had the right dose, we said we're using this dose. It was a guess. We didn't really know what time to treat patients, but we said we're going to use this time interval, and that was really a guess. In terms of the patient population, again, we sort of guessed at that, and we kind of went forward. I think we got lucky, but I'd much rather not be lucky in the future. So is the model broken? Let's go back to preclinical testing to start. So as somebody who does clinical trials, I consider myself to be a customer of preclinical work. And what I see is lots of problems. Usually preclinical work is done in single labs with individual models. They don't compare different agents from other trials. They're often not blinded or randomized. Their statistical methodology is really obsolete or really inadequate. And sometimes the models and the outcomes aren't clinically relevant. So, if you're not doing the stuff right in a preclinical model, it's going to be hard for us to do it right in the clinical models. In phase 2, again, we have inadequate data to know what are the most promising agents. We usually only evaluate one agent at a time in a phase 2 trial. Sometimes the outcomes aren't clinically relevant. Sometimes the study designs are not comparable for different agents. So somebody's, again, using drug A, B, and C as an example. Somebody does drug A and they're using infarct volume as their final outcome. Somebody's using drug B and they're using modified Rankin scale as their outcome, and etc. Different outcomes, different inclusions and exclusions. The study designs aren't comparable for different agents, and then we often have a best guess for dosage, duration, time of treatment. These trials, as I mentioned earlier, are underpowered for efficacy, and at the end of a phase 2 trial, you really have no ability to predict the possible success of this agent or device in phase 3, and that's a problem. I mean, you would like to have some idea going into a phase 3 that in fact you have such and such a likelihood of being successful in phase 3, and we end up guessing. So in the phase 3 trials, what do? We continue to make mistakes. We evaluate one agent at a time. We don't know if it's the right agent. We use a fixed trial design. Even though there's some things we're still not sure of, we say we're going to do it with this dose, this way, at this time, and even though we're not sure, sometimes we're studying the wrong patient population. And as I mentioned earlier, we have a very high failure rate. How about 100% failure rate in terms of neuroprotection? So is the model broken? I think that it is. I think that we compound our errors by our outmoded process. I think sometimes we're choosing the wrong agents to test. I think we're failing to compare multiple agents. I think very often we're not getting the right dose, the right duration, the right timing, or the right patient population. And so the worst part about all this is that some of these drugs that have failed for neuroprotection, I'm not at all convinced that basically they don't work, because a lot of the problems we might have is not really a failed trial, but a failed design. And I'll use TPA and stroke as an example again. You know, there were other trials looking at TPA and stroke at the same time we were doing that with the NINDS trial. They all used a different duration for treatment than we did and a different dose. Our study showed that TPA worked, their studies showed that it didn't. If we had used a different dose, if we had used a longer duration for, for entering patients or whatever, we would have had a negative study too. And then we would all be sitting here saying, you know, it's really horrible, TPA just crashed and burned, but, you know, really nothing works to open up arteries in acute stroke. And that would be the boat we're in right now. And we're not because we were smart, but because we're lucky. So what's the future? Really think we need to redesign the way we do preclinical research, phase 2 and phase 3. In preclinical research, we need a lot closer collaboration with clinical investigators, and clinical researchers need to help with the design. There's been a lot of traction recently on using multi-center preclinical trial networks. This is something that is being started in Europe, and the NINDS has been interested in this as well, in using blinding randomization and using modern methods of data analysis, very similar to what you would do in a clinical trial. And then development of ongoing trials with different experimental models. So in other words, you can have models that keep testing different agents as they come up. We think that in a lot of these areas, using adaptive designs can be beneficial. What we really need to do, typically in the preclinical research, you study one thing, you use your model, you say it works, and then you go on to the next thing. What people never do, or rarely do, is actually test and compare individual agents and combinations. So if you were studying neuroprotectives A and B and C, it would be really nice in these multi-center preclinical trial networks to look at A alone, B alone, C alone, A plus B, A plus C, or an ABC, to see what's the best individual or combination agents that work before you move on to the clinical arena. And we also need better dose and duration finding. So phase 2 process in human trials, again, will be informed by more adequate preclinical designs. I feel that adaptive designs could be very helpful in this phase for sure. And again, doing the same kind of thing where you're having ongoing phase 2 trials where you can basically look at different drugs or combinations, and when you get to a point where you've got a decision rule that says, okay, right now we're at the point with this drug or combinations that we have a 60% chance of succeeding in a phase 3 trial. You take that out of the phase 2, you do a phase 3 trial on that, and then you start studying other things in phase 2. And then you also weed out the losers. You get rid of the things that don't work, you kick them out, and you just don't go further with them rather than going all the way through to phase 3. And then better defining dose and duration. And this sort of is something that the cancer people, I think, have been doing better at than we are. And I'll see if I can get this thing to work. So this would be a traditional trial here. So what we would do, this is the example, drug A, drug B, drug C. And so what we're doing in phase 2 is we're looking at a control group, a drug A group, and we're seeing whether they work. And if that looks like it works, then we try to do a phase 3 on drug A, do the same thing with drug B, we do the same thing with drug C. What we're saying we should be doing in phase 2 is that we should be looking, we've got a control group, we've got a group that's getting A only, B only, C only, A plus B, B plus C, and there probably should be an A, B, and C as well. And then what happens is the less successful arms you kick out, the more successful arms continue in phase 2, and then those get kicked into phase 3. So if the drug gets promoted to phase 3 using this method, it's usually going to take about over 1,000 patients and 3 years to determine if it's safe and effective. If you're using this model over here, it's much, much less. And not only that, but you know you're going to have a much better chance of succeeding in phase 3 when you finally go forward. So, future of neuroprotection, I think that many people who do neuroprotective agents, and certainly the pharmaceutical industry, I think most of the big pharma companies have said, yeah, this goes nowhere. And they've got a sense of nihilism, and I think a lot of clinicians do as well, and it's because we've had failed translation. I think our preclinical and clinical trial testing have had very significant shortcomings, and I think that we need to do a much better job screening agents and identifying promising agents and combinations. And basically redesigning our entire approach using more flexible trial designs. And I think we probably even need to consider retesting failed agents using this new approach. And that's sort of a scary thought, but you know, there's lots of agents out there that have been used in the past that may actually be effective if we did them right. So, that was a lot of talking in a short period of time, and I know I don't talk slowly. So I would be happy to answer any questions on any part of the talk. But thanks very much for staying with me. Anybody wants to raise their hand, or you could type in a question, either one. Tell us more about anticipated regret. So anticipated regret is sort of a little tool that we're using in clinical trials, and I sort of alluded to this a little bit earlier, that I think it's a great tool that when you think you have a design that works pretty well, to go back and say, and your trial fails, is to go back and say, what do I wish I had done differently? Okay? And thinking about what that regret would have been and anticipating what that regret might be, you can have a better insight onto what your areas of uncertainty are. Any other questions? Not one comment, it looks like. Okay. The role of NET, NeuroNext, and NINDS interactions and relationships unclear. Well, that's a great question. So what let me issue a disclaimer in that I am not speaking for NINDS. I'm speaking from my understanding as being the PI of NET in terms of the way I understand the relationship. So the NET is a network that strictly does studies on neurological emergencies. We do phase 3 trials, and we do studies that are looking at interventions that occur in the pre-hospital or the emergency department phase of care. So it can be anything. It can be seizures, strokes, head trauma, meningitis, or whatever. And as long as the intervention takes place either in a pre-hospital phase or the ED, then it's a NET study. NeuroNext is really more of an early phase network that crosses the entirety of neurology, with the exception of stroke. And so NeuroNext will evaluate, anything else, movement disorders, neuromuscular disease, et cetera, usually doing early phase trials, not late phase trials. NET does only phase 3 trials. And you didn't mention StrokeNet, but I will mention StrokeNet. So the third network with NINDS is StrokeNet, which was just formed. So StrokeNet is responsible for stroke studies across the spectrum of stroke, so prevention studies, treatment studies, and rehab studies. They will do both early phase, phase 2, and phase 3 trials. If there are phase 3 stroke trials that'll be done by the StrokeNet, typically the net would be helping with them, so we would do those studies together if they're phase 3 trials. And if they were phase 3 stroke trials that only had a pre-hospital component, so like the FastMag, then that would just be done in the net. So I hope that. And the relationship with NINDS is that NINDS funds all these networks. So I'm not sure if that's entirely clear, but again, that is my own opinion, not those of NINDS. So Dr. Scott asked, how receptive are major peer-reviewed journals to results from adaptive trial designs? I think that's a good question. We haven't studied that specifically. We're actually looking to do a study to evaluate some of those barriers that might be there in major peer-reviewed journals. So
I'll address that a little bit during my
next presentation. At this time, Philip, how those will be received. There have been some adaptive trial designs that have been published in the literature. I don't know that there have been major issues, but it's certainly something that's still a little bit more up in the air, I think. And Will, if you want to chime in on that, feel free. Will is typing something now.
I'll address that a little bit during my next presentation.
Okay. Got another question coming here. Okay. In your experience as a mentor, what tend to be the problems with junior investigators designing trials? Well, I think there's sometimes a couple things that can tend to happen. I think a lot of times, I think a junior investigator, you're starting, you've got your first sort of agent or whatever idea that you're going with, and you're obviously very excited about it, as you should be. I think what happens sometimes is that you tend to become very biased, and you get too close to the things that you're doing, and it's hard for you to maintain perspective sometimes. And I'm not talking about you particularly, I'm talking about me and other people I've known, that sometimes you get really hooked with an idea, and it's sort of like sometimes you get the idea that you're not doing this clinical trial because you really want to learn if this treatment works. You want to do this clinical trial to convince all those idiots out there that it works because you already know that it works, right? And so I think that can be certainly one of the problems, and not having enough equipoise. And sometimes I think you can tend to get too close to things. I think the other problem can be that again, is not looking at all the other areas in which you might be uncertain, and trying to model some of the things you're doing on what you've seen has been done before. A lot of times what's been done before is not very good, and so you can end up going up the wrong track there too. Okay. So asked to discuss the approach of trial selection in the net, specifically slow recruitment. Well, that's a good question. So the trial selection, there are really two separate issues. One is what do you do with slow recruitment? The other is how do you select trials? So selecting trials within the net, I think we try to get good ideas no matter where they come from. So anybody can design a clinical trial that can be done in the net. You don't have to be part of the net to design a clinical trial that's done in the net. And so we try to go as broadly as we can to find the best people to do the best trials, and then put together a team of people to design these things. So it's not one person sitting in a room or two people sitting in a room trying to come up with a good idea. Well, then we also try to look at where we have holes in our portfolio. So, for example, a serious neurological emergency is spinal cord injury. The net is not currently doing any spinal cord injury studies, so that's an area that we've targeted that we would really like to be doing a clinical trial in. So we sort of look for areas where we think where we've got holes, and if there are good candidates for doing studies in that area, we would like to do that. Slow recruitment caused by competing trials can certainly be an issue. One of our requirements in the net is that every site that's enrolling in the net has to tell us whether they have competing trials, and they also have to tell us how they are going to manage the bias. So if you've got two clinical trials looking at ischemic stroke, you have to say how you're going to consider one trial over the other, and what's your method for doing that. So it's got to be a method that doesn't give you bias in terms of your enrollment for the trial. So I'll use this as an example. Maybe you have a stroke trial that's treating patients out to 6 hours. You have another stroke trial that's treating patients in under 3 hours. If your strategy is, well, we're going to do everybody in 3 hours into the 3-hour study, and the only people we're going to study in the 6-hour study is people who don't fit our 3-hour study, that really, if I'm the 6-hour investigator, that's a real problem, because that means I have no early patients that I'm getting from you and your site, because they're all being sucked off into another trial. If you had a strategy saying, well, we're going to do, even odd days. On even days, we'll consider the 3-hour therapy first, and on odd days, we consider the 6-hour therapy first. That's fair. But there's just got to be a method for doing that. And as far as with slow recruitment, I mean, the way you usually deal with slow recruitment is to, try to work with the sites and see if there's some kind of problem. Are they really up 24/7? Or the things you can do, you can help them. And once you get beyond that, it's essentially adding more sites. So hope that answers your question. Other questions? Within a single center study, slow recruitment. Yeah, slow recruitment is a big problem, and you just have to look at all kinds of things. You have to look at, and this is one thing that you have to look at when you're designing the study. If you design the study in such a way that it's very difficult to do, just in terms of the steps involved to entering a patient. If one step means you've got to do something in the emergency department, step 2 you have to go run off somewhere else to do something, step 3 you're back in the emergency department. If it's a complicated process for entering a patient, it's going to be very difficult to enroll. So trying to streamline the whole process and make it really simple for the people who do it. Sometimes there's an identification of patients problem, and that's something, again, you have to figure out, where are these patients and where do we identify them. And there can be a lot of issues. I think you basically just have to get somebody who is very good and has been successful in enrolling clinical trials, and do a really careful analysis of the entire process. And sometimes in a single center, you just may not have the patients. We always joke and say that the best way to cure a disease is to try to do a clinical trial on it, because then you don't see any patients with that disease anymore. But it's certainly a major problem. If you look at NIH-funded trials right now, 70% of them are under-enrolling. So it's not exactly a unique problem. Other questions? Okay. Will the NET coordinate a study without NIH funding, say funded by the pharmaceutical industry? Yes. We do have a policy in the net. We have an operating procedure for how we would do that. And if pharma would have to ask us to say, very broad terms, we have agent X and we want to study agent X in ischemic stroke, and specifically, we think, if they have some idea of who it might work in or whatever, just to give us an idea. What we would do that is we would design the trial and pick the primary outcome, do all the statistical analysis, come back, present that to pharma, and say this is how much it's going to cost. And if they want to fund that, then they can fund that, and if they don't, then they don't. What we would not do is let them design a study, give it to us, and say, will you do this? Because we would not do that. Like Will is coming up with one more thing. Okay. Anybody else? Other questions? Okay. There are none. Oh, got one more thing coming maybe. Wait. Keep waiting for this.
So after we get through the questions, I'm briefly going to go through how the course is going to be configured. And I think at this point, perhaps that, I think any other questions for Dr. Barsan can be emailed to him. So at this point, I think we will close.